Okay, it's 8 o'clock. We'll get started. Good morning and good afternoon, everyone. Welcome to Mission Bio's virtual event, Do More Single Cell with Less, Tapestri Platform Expansion for Hematologic Malignancy Research. We're going to share some exciting new updates on the Tapestri platform. Joining us today, we have our speakers, Dr. Koichi Takahashi, Associate Professor at the University of Texas MD Anderson Cancer Center. And we also have Dr. James Flynn, Senior Product Manager at Mission Bio. A couple of reminders before we start. We're currently recording this session and live streaming on YouTube. And throughout the presentations, if you have any questions, please enter them into the Zoom chat or the Q&A box at the bottom of your screen, and we'll answer them during the Q&A session at the end of all presentations. And with that, I'll hand it over to Koichi. Please go ahead.
Do you see my slides okay?
We do.
Yes.
If you want to actually put it in the presentation mode.
Oh, it's still not in the presentation mode. Okay, give me 1 second. I think I need to share this screen. How about this?
That's great.
Okay. Thank you, MJ. My name is Koichi Takahashi. I'm a physician scientist at MD Anderson, and today I'm talking about the clonal heterogeneity and the clonal evolution in AML, which are mainly get revealed by this Tapestri platform. So just a quick outline about what I'm going to talk today. I'm going to go over some of the background about the clonal heterogeneity overall in cancer and also AML, and the recent methodological advancement in measuring the clonal heterogeneity, and especially talking about the single-cell DNA sequencing technologies, and also biological implications of revealing these clonal heterogeneity and evolution in AML, and finally, some of the clinical implications of understanding clonal heterogeneity and evolution in AML. So this is really not needed so much, but there's an important distinction between the intra-tumor heterogeneity versus intra-tumor heterogeneity. So these DNA sequencing effort in large cohort of cancer patients, including AML, had really revealed a number of driver mutations that is involved in these cancer development and leukemia development, which also revealed this remarkable intra-tumor heterogeneity, or in other words, the heterogeneity of each patient, that each patient have a different genetic profiles. And this understanding that what genetic profile that your patient has a huge implications in terms of the treatment decision making these days, especially if your mutations is actionable. However, also in a single individual patient, there is also a heterogeneity exists inside your patient's tumor. So if you take a bone marrow biopsy from your patient, actually by morphology, all the cells look similar. They all look very, the same. However, if you analyze each single cell carefully, you actually notice that there is a lot of diversity and heterogeneity among each cells, which can be genetic heterogeneity, or phenotypic heterogeneity, and also could be epigenetic or transcriptomic heterogeneity. And today, I am mainly focusing on this genetic heterogeneity across each individual leukemia cells. And today I don't talk much about this, but there's also an increasing interest in revealing this spatial heterogeneity in your cancer cells. And this has been actually not investigated very well in leukemia field, but this has been increasingly well understood in the solid tumors field as well. And why is it so important to understand this intra-tumor heterogeneity or the clonal heterogeneity of your patient? because there's both biological and clinical implications by understanding intratumor heterogeneity. Actually, your bone marrow or your cancer specimen is a very diverse population of different cell state, which consists this tumor ecosystem. And understanding this tumor ecosystem has increasingly important to understand also how your actually tumor environment is created, including both the tumor cells and also the microenvironment, and how they respond to your treatment, especially the immune-mediated treatment. And also, clonal heterogeneity or intratumor heterogeneity is a consequence of the evolution. So understanding or describing intratumor heterogeneity accurately, as much as accurate you can, you actually are able to also draw inference on how the tumor got evolved. And this also has a strong biological implication how your cancer or the tumor developed through the understanding of this intratumor heterogeneity. From the clinical side, describing the intratumor heterogeneity or clonal heterogeneity has strong implication about predicting the therapy resistance, and as well as the overall clinical prognosis, which has been previously shown in the multiple publications. There are several or multiple methods to measure intratumor heterogeneity well, and most classical way is by morphology. Even though each cell may look like similar, if you look very carefully, pathologists has actually noticed that each cell look a little bit different by morphology. So hematopathologist or the solid tumor pathologist has historically been describing these morphological heterogeneity, which can be just observed by the microscopic evaluation. Flow cytometry is also a single cell analysis tools, which can immunophenotypically describe the heterogeneity across your samples. And recently, we are increasingly using this molecular platforms or the single cell platforms to describe this intratumor heterogeneity, which is the focus of today's talk mainly. The next generation sequencing actually really initially revolutionized the way of describing this intratumor heterogeneity. Although it has some limitation, it does decent job of describing intratumor or clonal heterogeneity. You have an AML sample in front of you, you smash it and extract the DNA from the specimens, and then you perform the NGS sequencing, which gives you this information about the varying allele frequency of each mutations. For example, DNMT3A mutation being 40% VAF, followed by NPM1, FLT3, ITD, and NRAS. And just by these information, there are a number of computational algorithms that will help you deconvoluting this clonal architecture or intratumor heterogeneity. For example, like a PyClone or Cyclone or Expanse algorithm. These algorithms all help to deconvolute these bulk NGS data and try to identify or try to kind of tease out how many clones or subclones you have in your specimens. And this is kind of the process where it's a lot of times kind of referred to as blender theory or the smoothie theory, where you have original ingredients of fruits which you blender. It's like smashing your samples and extracting the DNA out, and then perform the NGS sequencing is like trying to understand the ingredients of that smoothie. What is the percentage of each ingredient? And again, this type of bioinformatic or deconvolution process does a decent job in identifying the clone numbers or identifying the clonal structure of your tumor. However, the bulk sequencing can do as much as they can do from the data itself. And although you can actually try to understand the hierarchy of your mutation composition and draw the inference of what is the hierarchical structure of your tumor, you can almost never, unless you have a longitudinal or multi-site sequencing data, you can almost never really tell the most kind of important subclonal architecture of your tumor specimens. More specifically, whether your subclonal mutations are in a branching structure, or is it truly in the linear structure or not. You can probably tell that if you have a longitudinal specimens. However, if you only have a one time point specimen, this is extremely difficult to understand if you only have a bulk NGS sequencing data. So the idea was to, why don't we just sequence individual cells one by one? And this sounds a very simple and very attractive strategy by conception. However, there is many technical challenges associated with this single cell DNA sequencing. First is how to isolate each single cell. Traditionally, flow cytometry has been used to isolate each cell individually. This is labor intensive, and unfortunately, it's low throughput because you essentially manually sort each single cell one by one. Recent platforms, such as this Mission Bio platforms and other devices have increasingly using this microfluidic device to isolate each single cell, which helps the throughputness, but also have some kind of unique limitations like the doublets or the multiplets. Quantity of the actual DNA content in each single cell has been also the challenge from the technical challenge. Each cell technically has only 2 copies of DNA, unlike RNAs where multiple copies exist in each single cell. There's only 2 copies of DNA, so amount of DNA, absolute amount of DNA is just too small for sequencing, for the single cell sequencing. So therefore, historical method had inevitably had to use this genome amplification technique, like a whole genome amplification technique, which has lots of false positive issues or the false negative issues from the allele dropout, which is summarized in this excellent review by Nick Navian in Molecular Cell 2015. Also, this is somewhat unique to the single cell DNA sequencing, which is an accessibility issue, that DNA is tightly compacted inside a chromatin in the nucleus, and accessing DNA for each single cell has been technically challenging compared to RNA, which usually is in the cytoplasms. Therefore, there has been a number of the historical studies using the single cell DNA sequencing in AML. However, those studies have some limitation in the cell throughputness, or also require whole genome amplification, which kind of have introduced these artifacts or the false negatives. This is not the exclusive list of all the historical studies, but showing some representative studies of single cell genotyping in AML using the conventional methodologies like flow sorting isolation, followed by whole genome amplification or the amplicon amplifications. And as you can see here, some studies have limitation in the cell throughputness or the number of genes that they were able to interrogate because of these limitations. And these limitations kind of led to this historical nature that single cell RNA sequencing has been commercialized earlier than the single cell DNA sequencing, because there is this unique limitation in the single cell field about the accessibility and also the low quantity issue about the single cell DNA, that single cell RNA sequencing was technically more easier to commercialize. And therefore, it was no surprise that the 10x Genomics or the other platforms got commercialized earlier than the single cell high throughput platform like a Mission Bio. However, we were extremely fortunate that we were able to collaborate with the Mission Bio at early time point, where they had developed this platform which enables this high throughput single cell DNA sequencing. We published this in the Genome Research around 2018, where we went over the technical principles of how we make this happen to improve the accessibility by adding this protease K into this protocol, which dramatically improved the access to these genome site at single cell level. And with their molecular barcode and the microfluidic devices, the throughputness of this platform was dramatically better compared to the historical platform. For example, in this table where we are showing that with each run, we were able to genotype roughly 5,000 to 7,000 cells for a number of driver genes. And using this platform, now that there are 3 and even more publications out there using this platform to genotype AML samples in a large scales. These 3 papers and other papers more or less show very similar results. So I will take a liberty of just introducing our own studies, but please read other group studies, which really remarkably show the reproducible and very similar results among all the studies. So in our studies, we performed a large-scale unbiased single-cell genotyping of AML of about 123 AML patients samples. We initially used the 19G panel that Mission Bio had commercialized in the beginning, and also we later time we developed our own custom designed panels, which involved which covered the 38 genes of the 38 driver genes in AML, using this 290 amplicons. And since this technology was relatively new, we thought that we needed orthogonal validation of these mutation calls and also the zygosity calls. So we concurrently performed the bulk NGS or the ddPCR to validate each single mutation that we detected from the single cell platform. And also we performed the SNP array in the selected samples to validate some of the zygosity calls. Some of the quality control metrics using this platform, I really want to say that this platform is a very highly reproducible and extremely produce kind of a very good quality data. And each time when we run, the data is very reproducible. In median, from our experience, we were able to genotype roughly 6,000 cells per run, which I know with the updated platform now that the cell throughputness has improved, which we may hear later from James. In our experience, we were able to do about 48x depth per amplicon per cell, which we think is a very good sequencing depth as a single cell DNA sequencing. Again, technically each single cell only have 2 copies, or if you do a 48x coverage, technically you are sequencing each allele 24 times, which we think is a very good sequencing depth for a reliable genotyping. And there is a very good VAF concordance between the bulk sequencing variant allele frequency and the single cell DNA sequencing variant allele frequency, which tells us that even though you're only sequencing 6,000 cells from your sample, it is a good representation of your entire bulk samples. And estimated allele dropout from that platform was around 5.8%, which was much better than the historical whole genome amplification involved platform. And what was gratifying to see was that there was 100% concordance between, or 100% of the mutation that was detected by the single cell DNA sequencing were validated using either the bulk sequencing or the ddPCR when you use the cutoff of the 1.0% cellular population. And in fact, the single cell DNA sequencing detected more mutations, especially in the FLT3-ITD compared to the bulk NGS. And bulk NGS has notoriously been bad, especially if you use this capture enrichment system for detecting the ITD mutations. So this was something that single cell DNA sequencing did better job compared to the bulk NGS when you use the capture enrichment system. And we also later learned that if you do the 250 paired end sequencing, you have better detection of FLT3-ITD, which is some technical part, but compared to 150 pair end, it does a much better job of detecting ITD detection. And when you run these single cell DNA sequencing platform for each sample, you get this type of heat map for each sample, which tells us what is the mutation co-occurrence and the mutual exclusivity across all these single cell for your driver gene list. And we started to see this interesting observation that some of these functionally redundant oncogenic mutations are mutually exclusive at the cellular level. For example, in this sample where the FLT3 mutation and NRAS and KRAS, multiple NRAS and KRAS mutations were mutually exclusive at the cellular level, which was also similar in other cases, for example, this KIT mutation and the FLT3 ITD mutation, or this IDH1 mutation and IDH2 mutation in this case. And in this first case, where we also saw this mutual exclusivity between this FLT3 tyrosine kinase domain mutation and the ITD mutations. And all of these observations kind of inform us that functionally redundant mutations are mutually exclusive at the cellular level, which might implicate that actually having two or more mutations of the similar functional mutation, for example, this NRAS and KRAS mutation, might be deleterious to this, or it's not that does not provide a selected advantage to the cancer cells, and therefore it becomes mutually exclusive, or it's just sufficient to only have these type of mutation in one per cell, and therefore we see this type of mutual exclusivity at the cellular level. And now that you get this type of heat map at the single cell level, how can we reconstruct the clonal evolution pattern from these type of map? For that, we collaborated with the ETH Zurich people who had developed this algorithm called CITE, which was exclusively designed to infer the clonal evolution pattern based on the single cell genotyping data. And using this platform, we were able to reconstruct all of our samples for the clonal tree. And among the 123 samples, half of them exhibit the linear clonal evolution pattern like this, but while the other half of the patient had this branching evolution pattern like this. And among these branching evolution patterns, we also observed interesting cases like in convergent evolution patterns. For example, this case had a multiple parallel evolving clones, which essentially contains the same similar combination of the mutations like NPM1 plus IDH followed by FLT3 or MAP kinase pathway mutation. And this is similar to what other investigators have observed in other solid tumors, like in the renal cell carcinoma, where they have used this multi-site sequencing to identify this convergent evolution pattern, in this case with the SETD2 mutation in multiple areas of the mutations. And using this clonal inference of this mutation tree, we are now able to identify what is the order of the mutation that was acquired in your AML samples. For example, in this sample, we think that ASX1 mutation was acquired first, followed by SRSF2 and RUNX1 and IDH2. And understanding this mutation order at the precise level seems to be maybe important in AML biology too. And we got inspired by this paper in myeloproliferative neoplasm, where using the single colony sequencing technologies, they have identified two patterns in the myeloproliferative neoplasm, which start with TET2 mutation, followed by JAK2 mutation, or the vice versa, that you start with JAK2 mutation and then acquire TET2 mutation. And interesting that UK group have found was that depending on what mutation you started with, actually the clinical prognosis and also the clinical phenotype was significantly different in myeloproliferative neoplasms. So we also tried to see if this similar phenomenon might happen in the AML too, if the mutation order might dictate some of the clinical phenotype or not. In AML, this work turns out to be a little bit more complicated because there are multiple, many more driver mutations are involved. But when you summarize the initiating mutation, the number of the first mutation that started in your AML sample, most of the initiating mutations are the mutation that we expected as a clonal hematopoiesis, for example, DNMT3A or TET2 or ASXL1. But also in some of the AML samples, you actually start with these signaling mutations like NRAS or FLT3 mutations. And when you summarize the mutation order pattern in acute myeloid leukemia, at least in 123 AML samples that we have analyzed, there's a very diverse pathway for AML to have in terms of the mutation order. There is definitely, most likelihood mutation pattern exists, like DNMT3A followed by NPM1 or FLT3, which all know that is the most common kind of the trajectory. But that's really not the dominant feature of AML, that there is multiple other diverse pathways for AML to have when you analyze this mutation order in 123 AML patients. If you focus on these rare cases which started with the RAS mutation or the FLT3 mutation, at least we have observed that in these RAS first AML or RAS pathway first mutation AML, we saw that there is some increased monocyte counts in both bone marrow and also the peripheral blood, which might suggest that RAS first AML have a monocytic phenotype, which also goes along with the observation that in monocytic leukemia like CMML or JMML, the RAS mutation has been known to occur as a initiating event. So it seems like the understanding the mutation order derived from the single cell DNA sequencing might tell us some of these clinical phenotype as well. And this work probably will be augmented by this recent capability of DNA antibody-seq, which the Tapestri now is capable to perform this both DNA and antibody sequencing. We are showing in our paper some of the examples how these DNA antibody technology can help us understanding this genetic and a phenotypic co-evolution patterns. For example, in this case, where we understood this genetic evolution from the TET2, started with the TET2 initiation and followed by U2AF1, DNMT3A, and NRAS mutation, we were able to track the phenotypic evolution along with this genetic evolution. And from these information, we are able to maybe predict or infer what was the tipping point from the pre-leukemic to leukemic transition. In this case, we think this NRAS mutation acquisition really kind of drove this leukemic transformation from the pre-leukemic phase to the leukemia transition. And there's a multiple papers, I'm not showing everything here, but only the representative ones here, that is really making this field exciting, that this single cell multi-omics capability have really shown this understanding of co-evolution of the genetic and phenotype or even the transcriptome. In the last minutes, I'm going to go over some of the implication in the clinics using this type of technologies. Even with the bulk NGS, if you have a longitudinal sample, you can do a very good job of inferring the clonal heterogeneity and the clonal evolution patterns. However, if you have a single cell capabilities, you're able to also infer this clonal evolution pattern in much more granular way. For example, in this sample where we were able to tell which precise clone was responsible for the AML relapse in this setting with the treatment with the azacitidine and sorafenib, where we showed this FLT3 D835Y clone was pre-existed at the 1.7% at the diagnosis, which was clearly selected with this sorafenib treatment and later constituted the relapse. And this is very consistent with the in vitro evidence that D835Y clone is resistant to sorafenib, while other clone like D835E clone or ITD clone are sensitive to sorafenib. And in our patient samples, exactly the same thing happened, that D835E clone or ITD clone was clearly effectively eliminated by the sorafenib treatment, while this D835Y clone was selected with this treatment. And even in the primary refractory patients, where your blast is 80% from the get-go, and after the treatment, it's still 80%. So by just morphology, there's really no change. However, inside your bone marrow, there's a lot of clonal dynamics happening if you do the single cell sequencing, and you can understand the treatment responses at the clonal level, where you will learn a lot from even with the primary refractory patients, where certain clones are being treated or the certain clones are being selected with your particular treatment. And this type of capabilities have been increasingly applied to the targeted treatment context, where, for example, in the FLT3 inhibitor context or IDH inhibitor context, where the selection of these RAS mutated clones seems to be the consistent feature of these type of targeted therapy agents. So in future, we're envisioning the field where we are able to predict the response to the treatment at the individual clone level, and by the composition of what clones your patient have, you may be able to predict what treatment your patient may become sensitive or become resistant to that. And just a summary, single-cell DNA sequencing is a powerful tool to determine the clonal architecture, mutation co-occurrence, and also the mutational history. And potential clinical application includes this evaluation of treatment response at individual clone level and clonal mechanism of drug resistance. We couldn't touch so much base, but we are also particularly interested in the application of these technology in MRD setting, which I'm pretty sure in the next few years that these type of application will be published. I wanted to note that clonal heterogeneity is still underestimated with these type of platform because this Tapestri is a targeted platform. We definitely need a broader coverage of genome while maintaining the thoroughness in order to understand the clonal heterogeneity at a broader level. There's an exciting future opportunities with the multi-omics capabilities with the site-seq or the DAP-seq, and also the DNA RNA seq. I only touched the surface of the DNA antibody seq today, but I'm pretty sure you'll also get an update from the Mission Bio group how this DAP-seq capability is expanding later on. With that, I would like to end here, and thank you so much for listening.
Great, thank you, Koichi. At this point, I'll hand it over to you, Jim. Let me share that screen back.
Thank you, Koichi. It was a great talk. Very good overview of the platform. I just want to spend a few minutes talking about where Mission Bio is and where we're headed. So just an overview of the company. I think you heard it from the talk, is that really our goal is to move precision medicine forward. We're really interested in having a single cell platform that can unlock single cell biology and help the clinicians understand exactly what's going on with a patient sample and guide the therapy and understand the resistance to different treatments that are out there. So if you go to the next slide. This is now just looking at our company as a glance. We've sequenced millions of cells at this point. So we have a tremendous background of different cancer genomes that have been sequenced. We're well over 34 at this point. I think that was a number from last year. We're into the hundreds of custom panels that we've created for different customers. So there's different applications, different uses of the platform. We can create custom panels for that. We have roughly 800 users on our portal that are using it, and we're over half of the cancer centers in the United States have adopted this platform. We're also internationally, we've significant adoption in Europe and Asia as well. Go to the next slide. Just some very high level specs on the platform. What we can do with our Tapestri platform is single cell DNA sequencing. As you've seen, we can profile via targeted amplicons anywhere from down to just a few amplicons and targeted panels up to about 1,000 amplicons across the entire genome. So you can get hundreds of loci that if you're particularly interested in detecting variant changes, copy number changes that happen, rearrangements that happen in the genome, we can do all of those. We can design a panel to do that. Our output is on the order of thousands of cells that get there. So that getting thousands of cells out, you can enable detection of rare clones down to 0.1%. So very few cells that you can detect out of a large population of cells. And we are compatible. I think there was a question in the chat. Can we work with cell lines? Yes, we can absolutely work with cell lines. We are currently enabled to do human, mouse, or if you have a genome set, which is not standard, we can work with that as well. So please do talk to us if you have a project in mind. Go to the next slide. I think we'll talk about a couple capabilities in the platform. We, as I think Koichi pointed out, there's a great review article that was written by Nick Navin's lab about the things you can do with single cell sequencing. One of them being, can you detect the clonal architecture or substructure of a sample population? yes, you can do that. You can start to understand the co-occurrence of particular mutations that exist within a sample. You can start to do things like reconstruct the clonal lineage, which I think you just saw in the talk, which was fantastic. You can start to understand the acquisition of mutations as they progress with the disease, and as they evolve through tumor evolution. And all of that made possible by being able to, on a cell-by-cell basis, detect mutations and understand exactly which ones that co-occur in the same subsets of cells. So if you go to the next slide. What we've done recently, these are just a couple of updates on the platform, is we've heard customers who have, certainly in AML and other blood disorders, you have access to a large number of cells. Where we're headed, where we're going, is that often you don't have high numbers of cells. So what we've done is we validated the platform down to 20,000 cell input. That does not impact the performance of the platform at all. So what we're doing is essentially expanding the range of input for the cells that go onto the platform, and that is not an issue. This is in anticipation of getting into different applications that require lower cell input. So we've been able to conserve precious samples on the platform. And if you go to the next slide, we want to talk about also, we do have catalog content, which is primarily in the heme space, but we do have a tumor hotspot panel, which is focused around hotspots around solid tumors, which has been available for a few years now. What we're now offering, and has been an expansion to our catalog offering, is the ability that we've had over 30, 40 publications, I believe, last year. Now we're taking some of those that the authors have given us permission, and we've made those available to other customers who may want to follow on in other indications. So we're launching an acute lymphoblastic leukemia panel from our collaborators at VIB, myeloproliferative neoplasms from Peter Mac in Australia, and myeloid clonal evolution panel from Ross Levine at MSKCC. In addition to that, we have virtual panels as well. So we have another of pre-designed panels that are in a number of different heme indications, which are available. That's a starting point in our design software, where you can start with that panel. You can customize it if you wish, or order it as is. And then finally, you can completely customize the panel that you want to utilize in our design software, which is available. It's free for anybody to use if they want to sign up and try it. And again, you can make a panel anywhere from 20 amplicons up to 1,000 amplicons on that software, doing it in human, mouse, or any other custom reference genome that you wish to use. You can go to the next slide. This is just a couple of slides to highlight these new published panels that we're showing. This is from a paper from last year from VIB that was published in Blood, and they were interested in acute lymphoblastic leukemia. And so they based this panel looking at all of the most common mutations that are present in COSMID, as well as other databases, and they've formulated this custom panel. So it is broadly applicable to anybody who's interested in ALL. And their main question was understanding the clonality based by single cell sequencing, and could they utilize that for MRD or minimal residual disease testing? If you go to the next slide. This is a different group, also published last year. As you heard, MPN, there's the acquisition of different mutations. What's the order of those acquisitions of mutations, and when do they occur? What are the real drivers from the transition from MPN to full-on AML? So this was a panel, they were looking at early stage MPN patients and which mutations did co-occur in those samples. If you go to the next slide. And then finally, this is a really focused panel on just the evolution of myeloid malignancies. We do offer an AML panel as a catalog offering and a myeloid panel, which has a much broader spectrum of mutations which are covered. This panel is a little more focused, a little more looking at just the clonal architecture and evolution of panels. Also to note this, as Koichi mentioned, we can pair the addition of antibodies to the panel. So if you look at this paper, this was one where they paired antibodies with the DNA panel to look at both DNA plus protein changes, so genotype to phenotype kind of changes in the panel, which you can do on the Tapestri platform. And if you go to the next slide, this is an example of the kind of data you can look at different compartments within the heme lineages. We offer a standard 45-plex panel, which covers all the lineage markers present in blood samples. And so you can assess the DNA mutations in conjunction with cell surface markers on there. What we also are highlighting right now is that we've grown our catalog beyond that 45-plex. We now have about 150 total seq-D antibodies that are available through BioLegend, who's our antibody partner. And what you can get with that is you can construct your own panel of antibodies to assess what's going on at the cell surface level, as well as assess DNA mutations at the same time. As well as if you have needs for custom conjugates, we can do that as well. So please reach out to us if you have a project in mind to do multi-omics on our platform. And I think I have one more slide just highlighting what I just mentioned, doing DNA plus cell surface markers. We've had a number of publications come out this past year, looking at therapy resistance, CHIP, clonal hematopoiesis, looking at MRD testing with AML, as Koichi mentioned. That's a focus area for us to understand what is the limit of detection in single cell. Can we assess very rare cell populations? So that's something to look forward to as we keep working on that. And then also looking at resistance and or clonal evolution of cancers in response to treatment is another area of focus for Mission Bio. So with that, I think I will turn it over. I know we have a number of questions, so I want to leave some time to get to that. So with that, I'll turn it over to MJ if we have some questions from the audience.
All right. Thank you very much, Koichi and Jim, for the wonderful presentations. As a quick reminder, if you have any question, you can type it into the Q&A box on your screen. And I'll see if we have any questions that came in.
While you're looking those up, I did see some in the chat. One question was, can we work on cell lines? Yes, we can absolutely work on cell lines. As I mentioned, the ability to work with any custom reference genome that you have. So if you have a non-standard cell line, we can certainly work with that.
Okay, I think a lot of the questions were answered directly in the chat box and the Q&A box. Let's see if there were any unanswered questions so far. Okay, this one is for Koichi. So what are can you talk about some of the next key questions to address using the single cell DNA sequencing technology?
Well, I think there are lots of key questions and that there's lots of things that we can do or we need to do. I think one of the limitations of using at least the current technology is, again, the targeted nature of the sequencing. So we, at least in our work and most of the published work using this platform, we are only covering the well-known kind of the driver genes. But with that, we can understand the clonal architecture and at the driver gene level, and also we can understand the clonal evolution of the driver genes, but not necessarily the entire kind of level of clonal evolution. More specifically, we couldn't infer the time component because we're only capturing the driver mutations. And in order to understand the time component, I think we also need to sequence like entire somatic mutations, including the passenger mutations too. And that is something that in order to do that, but while maintaining the cell throughput is extremely difficult task, but I think that is the next step that we are definitely thinking. That's from the clonal evolutional side. I think again we were very excited about the multi-omics capabilities, which could be either the combination of DNA plus antibody, or I mean RNA plus antibodies. The CiteSeq is also producing lots of excitement. And also this DNA and RNA, which are the next really the holy grail, I think the capabilities. I think there are published publications around that you can genotype from the RNA transcriptome, and there are definitely interesting data that is being published, but there's some limitations of doing that. So I think we are genuinely waiting for this pure simultaneous DNA RNA analysis as well. And I'm pretty sure these technology will definitely come along. And finally, the clinical application side, I think we are particularly interested in the MRD application of these platforms. And I think those things are the next key questions or the projects, which I'm pretty sure there are also a lot of others.
Great, thank you for those perspectives. Let's see. This might be a little bit more a technical question. This is one for Jim. The question is about if the oligos are attached to the heavy chain of the antibodies, and whether they could use the secondaries to find the dilution of the antibody that they're using for the multi-omics experiment.
Ooh, that's a very technical question. I can give an answer. I certainly can provide it if you follow up in an email. That's a question I'd have to follow up with our friends at BioLegend to see. Believe the tag is attached to certain regions, but I can't be specific. I do know that we try to only attach 1 or 2 tags to the antibody and then HPLC purify that. So please do follow up in an email. We can discuss if that's something of interest.
Okay. I think this might be actually a question for Koichi. So did you apply any cutoff, for example, a minimum number of cells that a mutation was detected from in order for the mutation to be considered as real?
We didn't apply the minimum cell number, but we definitely applied the minimum cutoff of the cell population, which in our case was 0.5%, because that was the lowest that we could verify using the ddPCR at least. But those are the minimum cutoff of detection. But if you just use 0.5% cutoff, there will be also lots of false positives. So filtering out these false positives will be the very important task computationally.
Thank you for that. This is actually a question for Jim again. Is there a way for me to participate in the published panels program? How do I collaborate with Mission Bio on that?
Yes, absolutely. As we get more and more publications out there, please do reach out to your local rep. They will put you in contact with us on the product team. We're looking for additional novel applications out there. So yes, that's definitely an option if people are interested in collaborating and developing panels that could be available to other researchers. We can do that.
Great. And then this is, I know Koichi, you sort of covered this in the presentation, but a question also came up. What are kind of your perspectives on the future of single cell DNA profiling going into the clinics, especially in the maybe in the near terms?
Yeah, that's a very complicated question, actually. I mean, we are definitely excited about the data that single cell DNA sequencing provides. It's scientifically super nice and it's beautiful. It's no question about that. But clinical decision making may not need that information at this point, at least. Or in other words, we decide the treatment decision, or I don't know how to describe this very well, but bulk sequencing may able to provide sufficient information at this point in terms of the clinical decision making, because knowing the subclonal architecture may or may not really determine the clinical decision making at this point. But that's just because our treatment is not that sophisticated enough. So that doesn't mean that we should not know the subclonal architecture in a granular level like this. And I'm pretty sure in 10 years or 20 years from now that we are using this type of technology routinely, because probably by then our treatment is more sophisticated and we need that level of information. At this point, I think bulk sequencing does, provides a good enough information most of the times. But so I don't know whether a single cell application of immediate clinical application needs. However, maybe MRD, we might have a little bit different opinion. I think MRD might be the most immediate maybe application that we can think of.
Great. I think this is a question for Jim. What is the typical cell capture rate?
The typical cell capture rate is really dependent on the sample quality. So obviously, if you have like nice, healthy cell lines that are at a homogeneous population, they'll have a capture rate closer to 10%. A more degraded sample might be on the range of 4 to 5% on the platform. It really depends on the quality of the sample.
I think we have time for just one more question that just came here for Jim. What's sort of next for Mission Bio? Anything that you can share that's on the horizon?
On the horizon, certainly we're going to be expanding our content around heme, but also into solid tumor, and you'll see some updates around sample prep, the ability to take solid tumor samples and put them on the platform, content with that. We're doing a tremendous amount of work in the cell and gene therapy space. Obviously, if you have cell material, what you are going to put into a patient, it's very critical that you understand the edits you make to those cells and the safety of that material. So I think you'll see a lot more about that from us. Other than that, Koichi mentioned some future looking statements around different analytes, which we might put on the platform. We have no announcements at this point, but that's certainly something to look for in the publications coming out on our platform in the future.
Great, thank you. There's one question that came in. I think we have time to just answer that one before we end. And I think it can be either Koichi or Jim for this one. How long can you preserve a sample before you sequence it for single cell sequencing? Koichi, do you want to take that one based on?
Well, we never tested how long it can last. So I mean, in our experience, it can be preserved after the library prep. It can be preserved, for a while, like a regular NGS library. But Jim, you might know a little bit more how longest you can test.
Yeah, that's a tricky question. Depends on the sample material. Depends on how it's prepared. Is it dissociated, not dissociated? Are we talking about cells, nuclei? There's a bunch there. If you have a specific technical need, I'm sure we can talk that through with our field applications team. Maybe be more than happy to work it out. But In general, if it's a biobank sample, we've seen that generally you can utilize them on the platform if prepared in a way that the integrity of the cell material is still maintained.
Great. I think that's all the time we have for the questions. If we haven't, for one able to get to your question, we'll follow up with you after this webinar. Thank you, Koichi and Jim, again for this wonderful webinar. And thank you everyone who attended today. For those of you who have registered for the webinar and stayed with us until the end, we'll be sending in a Uber Eats voucher. So please be on the lookout for an email shortly after this today. And thank you, everyone. And that ends this webinar.
Thanks everyone for joining.
Have a good day.